Hello, I'm Joe Landry, an assistant professor of human and molecular genetics at Virginia Commonwealth University and cancer researcher at VCU Massey Cancer Center. I'm also a grateful recipient of a V Foundation Scholar Award given by the V Foundation to support my research on studying oncogenes in models of human breast cancer. Thank you to the V Foundation for their support. In honor of the V Foundation's 20th anniversary, I will now pass the V to my colleague, Dr. Stephen Grant, who has twice received a V Translational Research Award. Dr. Grant is a professor of hematology-oncology at VCU, as well as the Associate Director of Translational Research and co-leader of the Developmental Therapeutics Research Program at VCU Massey Cancer Center. Dr. Grant?
Thank you very much, Joe. I'd like to take this occasion to express my profound gratitude to the V Foundation for their support of our research in the form of two separate translational research awards. My major focus of interest is in the rational combination of targeted agents in hematologic malignancies, that is, blood cancers such as leukemia, lymphoma, and multiple myeloma. Our underlying premise is that interruption of a single pathway in these diseases by targeted agents is often insufficient to achieve significant clinical results. Our hypothesis is that interrupting a second complementary pathway, which compensates for the effects of interrupting the initial pathway, will disable escape mechanisms and will result in more leukemia, lymphoma, or myeloma cell death. In our initial work, we, supported by a Translational Research Award, we investigated interactions between an agent known as flavopiridol, which is a cyclin-dependent kinase inhibitor, and which interferes with cell cycle progression, with another class of agents known as proteasome inhibitors, in this case bortezomib. Proteasome inhibitors are known to be very effective in multiple myeloma and certain forms of non-Hodgkin's lymphoma. We found highly synergistic interactions between these agents in the test tube. We're able to translate these results into two separate phase 1 trials for patients with refractory or relapsed multiple myeloma, non-Hodgkin's lymphoma. The results of these initial phase 1 trials have been very promising, and we are now working with the National Cancer Institute and the pharmaceutical industry to develop more definitive phase 2 trials of this regimen for patients with refractory myeloma in the near future. The second award involved investigation of an interaction between one agent known as sorafenib, which is a signal transduction inhibitor, which is known to be active in certain solid tumor malignancies, including renal cell carcinoma, but it's also effective in a certain subtype of acute myelogenous leukemia. And we found highly synergistic interactions between sorafenib and a class of agents known as BH3 mimetics. These are agents which interfere with the function of proteins that prevent neoplastic or cancer cell suicide. And based on our initial results, supported by the V Foundation, we've been able to obtain a 5-year R01 award, which will allow us to explore these interactions more definitively and hopefully to develop in the near future a clinical trial concept in patients with refractory acute myelogenous leukemia. In sum, we are profoundly grateful for the support of the V Foundation and wish them every success on the occasion of their 20th anniversary.